Okay, so today we will learn about human mesenchymal stem cells. So I'm at home right now and I couldn't get my graphics tablet, so I'll just use the laser pointer. And this happens to be from a professor from the university where I studied. I did MS in Denmark. It is now called as University of Southern Denmark or Syddansk in university. Anyway, so human mesenchymal stem cells happen to be something of great interest in stem cell biology. We haven't discussed it as much, but one thing it is nice to know that if there is huge success in clinical trials with stem cell biology or stem cell technology, majority of it is because of mesenchymal stem cells. So what are mesenchymal stem cells? They look something like this. They are fibroblast-like cells, and they are like cross-shaped or so the fusiform. And they do not have any hematopoietic cell surface markers like those of CD34, 45, and 14, but they have other markers, something like 29, CD44, 166, and others. We don't have to mug up those things. But we need to remember that there are majority, we can get them in large quantities from bone marrow, like I think we discussed it in the last class as well. And so they do have some origin or some lineage similarities with that of hematopoietic stem cells. Hematopoietic stem cells is kind of network that is present in all over our body in a way. You can get mesenchymal stem cells from any of these tissues, even in the adult tissue. You can get them from skeletal muscles, subcutaneous layer. You can take any of these things. Bone marrow is the major source I was just discussing. We can even get from deciduous teeth, the milk teeth we call, and also we can get from fat. When we see these sources, we understand something better or good is that we can get these mesenchymal stem cells rather easily compared to that of, say, embryonic stem cells or any other particular adult stem cell, because you have to harvest the organ in that case. But because mesenchymal stem cells are present all over the body in a way, in one sense at least, so they can be easy. Some of them can be easily obtained. Say deciduous tooth, we are anyway going to throw them away. Fat, like in some forms of liposuction they perform for individuals, it's almost like that. We can get those from there. Peripheral blood, we can just take the blood a little bit, like in maybe like as blood donation, and then do it. So basically, the person who is donating them is not adversely affected in some of the tissues at least. We can also get them from fetal tissues, but anyway, we are not as interested as is with the adult tissues, because we want to have patients, we want to treat patients, and majority of those patients are individuals and grown-up ones. So these are good things to look at. And not only that, there is also that the mesenchymal stem cell can become many types of cells, those that are involved with muscular system and the bones, okay, skeletal system. The osteocytes, chondrocytes, that's in the cartilage, adipocyte, that's the fat, myocyte, that's in the muscles. This is with tenocyte, is with tendons probably, and stromal cells. So they have a huge potential for treating problems with the skeletal system and muscles, etc. So if you look at the mesenchymal stem cell, it can, as I was describing, it can become many different cell types that are which will eventually give rise to the bones, or will we give rise to different types of cells? You can see also cardiomyocyte. When we discuss about the heart diseases, it will also help us understand a little bit about it. If you think about it in a way, as we discussed before, mesenchymal stem cells are a network of cells that are connecting majority of the body. That means it's not just isolated like a lung in one place or a kidney in one place. It's like a fluid kind of thing that is connecting different organs in one sense. So there are many different. Say, as I was telling you, there is an extensive network of mesenchymal stem cells in the body, and there is some people think that mesenchymal stem cells from one place to another place, or one can contribute to the other, and similar such hypothesis. But there is no exact proof that this is the case. Meaning, the people are still divided whether there is whether there is a network of tissues contributing to each other or not. When we try to study the genetic signature, we would find there are a lot of similarities, but there are also differences. In the sense, they are not identical. Different mesenchymal stem cells isolated from different tissues are not identical. If we claim that mesenchymal stem cells are identical, are all one network, then we would expect that there should be genetic signature. They should have been identical. But again, when we say genetic signature, if we are reading the genes, sequence is supposed to be identical in all the tissues, but that's not the case. Each round of cell division may end up having some differences. So we cannot really prove or disprove this hypothesis at this time. But to my understanding or my opinion, okay, you should all understand that majority of the papers that are given in our prescribed book are reviews. So there are some things that are completely established. There are many things that we don't know yet. And sometimes the author gives their opinion. It could be qualified, but it might need proof. In that way, I would think at this time that the mesenchymal stem cells are kind of network within the body. But it could be disproved too. Okay, how do we isolate mesenchymal stem cells? We'll just see a few things, one or two. One of the easiest ways that the mesenchymal stem cell adhere to the plastic surfaces, and the majority of the other cells may not. So if you let the culture of cells allow them to adhere and they wash away the rest, the majority of them are mesenchymal stem cells. But that's not a good way of doing it, because there is always contamination from hematopoietic stem cells. I mean, that is the mesenchymal stem cells, purportedly one that we have isolated using this technique, is still heterogeneous. So that's a primitive one, but it's not very ideal. The other one would be monoclonal antibody using cell surface markers that are specific for mesenchymal stem cells. It's STRO1, CD146, etc. So we take the bone marrow cells, then we can do cell sorting using these monoclonal antibodies, and then we can characterize and see what kind of mesenchymal stem cells we have. There are, if you go and search on the literature, you will find that people are even trying to isolate stem cells from even urine. That's what I heard one of our alumni sister was doing. So different labs are working in different avenues, and they're trying to identify different sources of stem cells and see their applications in stem cell technology. So we already discussed, peripheral blood, all these are good sources. Deciduous teeth is also one. And if we happen to, because people, there's deciduous teeth are just falling away anyhow, you can get them, take their cells, and then take the cells from the pulp, and then we can probably get some mesenchymal stem cells from there. Okay, so mesenchymal stem cells, as I was discussing, they are into three categories you can think. One is that of the bone, tendon, stroma, and cartilage. This is forming the skeletal system. And then the other one is the muscles, right? Skeletal muscle cells, maybe cardiomyocytes, and etc. And the other one is the adipose tissue. So these are the three categories that one can differentiate mesenchymal stem cells into. And it depends on what we are growing when we are growing them, what are we growing them in? So something like wind signaling is there. And one of the noteworthy one, yes, mechanotransduction. Unfortunately, I cannot draw. Majority of the time we discuss about cells responses or differentiation as a response to biochemical factors. We should also understand how the surrounding hardness or the mechanical forces could induce the cell differentiation pathways as well. I hope you might have studied in cell biology where they grow the same type of cells in three different consistencies of the same medium with gelling agent. The more hard the gelling agent, the bone-like they become. Right? When the gelling agent is not as much, with the lesser the hardness, then they become normal or other types of tissues. So the surrounding physical forces that the cell is experiencing will also affect the way the mesenchymal stem cell differentiate. This is also a kind of signal. So the source, as I was talking about tonsils, you just take these tonsils, centrifuge them, and then we can take those cells and put it on plate, and that is how we can get mesenchymal stem cells. The other one is from liposuction aspirates. I hope you know what is liposuction. They put a long rod into the body and try to suck out the fat that is under the skin, right? And that fat, adipose tissue that is collected, is wasted anyway. So from there, people have isolated the mesenchymal stem cells. And the other one, usual or more popular, is the bone marrow aspirate, because when we want from a patient, this is a good source of taking from the bone marrow of the same person. We will discuss a little bit about the terms about autogenic in a while. And then put it into, and then isolate the mesenchymal stem cells. And then we get many different types of cell types. I'm unsure about the nervous system, but it is likely as well. You can see some of the labs. If you try to search, when you are searching, when you're reading about some of these, if there are any web hits that are taking you to some lab homepages, go visit them, read about their work, and then you can probably also contact them for your future higher education and other things. Okay, there is something interesting. There is for the use of mesenchymal stem cells in regenerative medicine, there is one of the issues that we have to discuss as a part of stem cells in general. The in vitro cell growth or replicative senescence of human mesenchymal stem cells. There is a simple, nice story I would want to discuss, is that there is a Nobel Prize winning surgeon called Alexis Carrel. he proposed something like cells are immortal. You can grow them as long as you want, and they would keep on growing. And he claimed to have put those some of the fibroblasts to grow for 5 to 10 years, and some as long as 34 years. And that was something of a miracle in a way. How could that be possible? And later, people became suspicious of those results because nobody else was able to replicate them. And then they followed his experimental procedures. There must be a lot of errors. One of the errors is apparently giving fresh cells in the culture every day. So is that the same cells replicating over and over, or is it the fresh cells that were put into the culture that was growing? Um, the person, Alexis Carroll, apparently knew about it, but he never admitted it. As we keep progressing with fame and other things, it's very psychologically common that we all build a shell around us, and we are not um, vulnerable, or we show as if we are not vulnerable to mistakes. And anytime that happens, the best thing is to be able to say, yes, I made a mistake. Sorry. And that is always a good thing. So what happened was, this is the web page. You can go and try on Wikipedia. That's good enough. Hayflick, he took upon these experiments, and he was suspicious, as many people were. He tried to do the same experiment. And many of these times, his cultures stopped at a certain point. And then he went back and looked at why did they stop, when did they stop. All his experiments were stopping after, say, about 40 replications. So there is something about, based on his cell type, of course. So there is a limit to how many times a cell can divide. Population doubling, you can also say it as. Or that is also replicative senescence. As the cell divides, they would stop growing. So the age of donor is inversely proportional to population doubling. PD is population doubling. So if we take mesenchymal stem cells from very young person, say a newly born baby, then the number of population doublings can be very high. But if we take from a very old person, then the number of population doublings is very less. That is because that is about replicative senescence. So normal human fetal cells, they would grow between around 40 to 60 times in cell culture. We are talking about in vitro, okay? So that is what is Hayflick limit. That is the number of times a cell can undergo cell divisions. That's about 40 to 60 times. And how is it relevant to what we are talking about in terms of mesenchymal stem cells? Mesenchymal stem cells, the older the person of the age of the donor, or the older the donor, the less number of population doublings. The limit is lesser. It'll be below 40 probably. For human fetal ones means very young, right? And they are having about 40 to 50 times. Then for an adult or an old age person, it will be maybe 20 times. So that is called as Hayflick limit. That is because of telomerase activity. And before I go on, I would want to
So,
also
I have a
explain.
doubt.
Yes, please.
Uh, is the division we are talking about, is it a successive stage or a mature cell dividing to grow its own population?
Um, so let me finish this, and then probably I will answer. I would have answered. Let's see.
Yes.
So what doesn't fall in replicative senescence is apparently embryonic stem cells. Majority of the other cells, okay, the adult stem cells also have replicative senescence. And the completely differentiated cells, they actually may not divide for long at all, maybe 1, 2, or 3. Terminally differentiated cells. The adult stem cells still have some stemness, and they can divide a little further. Embryonic stem cells, at least in theory, are the only cells that we know of that do not have replicative senescence, as long as the culture conditions are good. Okay. Rishi, did I answer your question, or can you tell me what your question is?
Yes, this doesn't take into account differentiation, just the division to grow its
Yes,
own population.
that's right. That's right. This is about only cell division only.
Okay,
And
sir.
again, of course, differentiation is going to affect. Terminally differentiated cells will not have this much of population doubling. Okay. Whereas stem cells can have a higher, and embryonic stem cells will have the highest in a way. Yes. Okay. And the reason for it is the telomerase activity that we have already discussed, I guess. But still, we can always try to understand it in this perspective. The chromosomes have telomeres, and telomeres are synthesized by telomerase. The embryonic stem cells, if they were there, they would have had highest amount of telomerase activity, because of which the telomere size would be larger, longer. And also, yeah, and as these cells are differentiating from, this is not differentiation, okay, here it is showing as population doubling. As the cells are differentiating from embryonic stem cells or ICM towards the completely differentiated cells, the amount of telomerase activity also goes down, because of which the telomere size will gradually keep going down and down. And there will be a stage after which the cell, the chromosome will start losing some of the genes, and because of which the cells will no longer be able to divide and they would have died out. Except in embryonic stem cells, which have high amount of telomerase activity, and similarly also the cancer cells, they also have telomerase, a high amount of telomerase activity. And when they, because of that, they can address the, because of telomerase activity, they do not have this diminishing telomeres. So they do not fall under the category, or they are not limited in terms of their population doubling, or they do not undergo senescence or replicative senescence. So I also brought this thing is majority of the times we will only stick to the knowledge part of it, and we want to learn what is it meant by. But it's also sometimes important and actually interesting also to know about stories that are involved with these findings. How did these people come to conclusions about it? What is the story behind it? You can talk about the story of double helix. You can talk like stab cells we discussed before. We also are discussing this now. And anytime there are problems with these kinds of stories where scientists have made mistakes, we need to learn those things and try not to make those mistakes. We need to learn how somebody has found out very nicely, accurately, and we need to find those. We need to find those ways as well, and try to inculcate those kinds of habits and thinking and integrity. There will be mistakes. Science is about mistakes, right? There will be mistakes because we don't have complete information about everything. It's an uncertain zone or area, and we are trying to find out. Only what I would encourage you all is never to get yourself, the ego, get in the path of knowledge acquisition or knowledge generation. That is the only thing we can control by ourselves. Willfully, we shall not do any crimes in generating knowledge or propagating the knowledge. Yes. Okay. And yeah, this is so. Even mesenchymal stem cells have these problems with replicative senescence. That's what we were discussing this topic about here. But it's highly relevant to the whole cell biology or stem cell biology. So people with old age are the ones that actually need the cell therapy. And if you want to get his or her own cells, and they are not able to, uh, or they're going to undergo replicative senescence too fast, then they are not useful to us. So in that case, how do we address the problem is the question here. So some people have suggested that we can overexpress telomerase gene, but then there is a problem of itself that can become cancerous, right? When you have high amount of telomerase activity and you're inducing um, continuous, or you're giving the cell with ability for continuous cell divisions, then it's like you're creating a cancer within itself. But anyway, they can go up to 250 population doubling because you overexpress it. There are problems, right? First, we are doing genetic manipulation. That itself is a problem. Second thing, we are giving a capacity of unlimited, kind of theoretically at least, or abnormal population doubling. And that is also a danger. But nevertheless, it is a strategy, and we need to learn the strategies. Because of continuous expression, there will be a problem. So people have suggested conditional overexpression, which meant that occasionally you have an inducer. And let me see if I can draw with the mouse. Say, for example, if this is a gene, okay, that's very crooked. And you put it under an inducible promoter. And this is the telomerase gene, assume that. Because if it is continuously expressed, there is going to be a problem of transformation. So what we can probably do then is to put a promoter that is inducible by a chemical component. Occasionally or intermittently, we give this inducer, so we can probably regulate the amount of expression of telomerase. That is one of the solutions that were provided. The other one is to use trichostatin A treatment, which will give transient expression of um, telomerase gene. I think this is better, unless if there are any other side effects of trichostatin, because these two involve genetic manipulation of the genome, and that is a problem. And I guess this one probably we can probably avoid, and we have to look at if the telomerase gene is under the control of trichostatin, or directly or indirectly. We should try to probably study it better. Anyway, these are not ideal solutions yet. We found some ideas. We have to work on them and refine them and see if it works. We'll keep it. Otherwise, we will discard the idea and find new ideas. So, as we discussed, mesenchymal stem cells have high amount of propensity to dividing, to differentiating into bone and other things. So if we want to do that, we can use BMP, that is bone morphogenetic protein. I hope you remember we discussed about BMP4 when we are talking about deriving gametes from stem cells. Or it is in the primordial germ cell one. There are many different BMPs, BMP4, 8, and many other factors are there. There is also another one called as transforming growth factor B, TGFB. When we overexpress them, they can differentiate into specific lineages. Mostly, they can become the osteoblast, something like this. If we have EGF treatment, then they differentiate into the bone tissues. And if we add PDGF, that is platelet-derived growth factor, they will remain in mesenchymal stem cell state for longer time without differentiating into something. So there is one more advantage of mesenchymal stem cells. There are two things. One is hypoimmunogenic, meaning that they do not arouse immune reaction from the patient, just like that. Because mostly it's because they express intermediate level of MHCs and class 1 molecules. So they are less and less detectable HLA2 class 2 antigens, because of which they do not arouse they arouse much of immune reaction from the host. So why we are discussing this is probably then we can use cells from one person to the other person, right? So immune reaction, immunorejection would be much lesser. The other term is immunosuppressive. The cells that we are putting into body can suppress to a degree the immune system of the host, right? One thing is they themselves are not highly reactive to the immune system of the body. Second thing is they are able to suppress the immune system to a degree um, of the host cell by interfering with T cells, alloreactivity, or with the dendritic cell maturation. The diagram here will summarize a little bit. We are not going into all of these details, but you can have a look at the PPTs as well. There is a notes also associated with it. The mesenchymal stem cells interact with several cells of the immune system. And they can, one second, they can interact, and they are also interacted upon by these cells. There are some of the cells can have negative effects on the mesenchymal stem cells, but the black lines indicate the effect of the suppressive effect of mesenchymal stem cells on these, and we also have some of these cells can have an effect on the mesenchymal stem cells. So mesenchymal stem cells, for example, they are inhibiting natural killer cell proliferation. They're inhibiting T cell proliferation, right? You can also see B cell proliferation and differentiation. There are many things that are actually being done. So mesenchymal stem cells have an effect on the on the immune system, and they are kind of the term that is used is something like immunoprivileged, in the sense they do not they are not acted upon by host immune system. And second thing is they can suppress a little bit of the immune system as well. So before we go into some of the things that have already been done, we'll just talk about what is autogenic, allogenic, xenogenic. This is the source of what is done. Autogenic is from the same person. Allogenic is from other individuals belonging to the same, we'll take it as species. And xenogenic would be taking from some other species probably. And why we are talking about this is if we want to give mesenchymal stem cells, okay, I'm unable to write with this, so I'll try not to use it. If we want to use mesenchymal stem cells as a solution for therapy, then where should the mesenchymal stem cells should come from? Should they come from the same person, or from another individual, or from other sources, any other sources than the humans problem? So best thing would be for autogenic, to get the bone marrow, and so that there will not be any kind of immune responses eventually. So that would be an ideal situation, but it's not always possible. First thing, replicative senescence is a problem. Second thing, the patient might already be suffering, and we cannot inflict more on this person, sometimes at least. Then getting allogenic is the best one. But the problem is that immune reaction is a problem. And allogenic, if you're getting from younger donors, then it is better. But it's not always easy for younger donors to donate mesenchymal stem cells. Xenogenic is highly risky. The supply is on our hands. We can take from other animals probably, but certainly it's not a There will be some incompatibilities with the organization of the body. And secondly, immune reaction will be severe. Okay, then in what state should we give? Should we give mesenchymal stem cells as such? Or should we differentiate the mesenchymal stem cell into something, the desired cell type, and then give? These are some of the questions that we already discussed when we are talking about human embryonic stem cells. As we said, you can give probably mesenchymal stem cells and in undifferentiated state, because maybe we are saying that it's a kind of network and they can move to different organs. And in that case, probably it's not a problem. We can't say the same about embryonic stem cells, right? And we can probably ideally differentiate and give so that immediately they will go to the uh, site of injury, and they will probably repair the tissue and hopefully help in the recovery of the patient. Second thing is site of injection. Should we give where the damage is, or should we just put it into bloodstream and hope that the cells will go to the areas of injury or deficiency, and they would repair the individual? Well, these two are almost what we were discussing, right? The should be local or systemic is about yeah, as I was telling you in the little bit earlier, if these mesenchymal stem cells have a network, and giving it systemic is all right, but local would be better, I guess. Okay, now we are seeing some of the solutions, some of the studies. It's a table of studies. We don't have to mug up all these things, but it will be good to appreciate a few of them. These clinical trials, okay? All of them are in clinical trials. This is for bone and cartilage regeneration. So there was a tibial non-union. They were tried on 20 different patients. The source of the mesenchymal stem cells is bone marrow. And what they have seen is about 15 of them, they have kind of the union has happened. The bone has got joined. Well, these trials are difficult to confirm whether is it working or not working, right? If it is working, we would have expected all the 20 people to have union of the joint repair or the bone joint. But what is the negative control is a question. Are you comparing with any other patient who has similar problems and we did not use these cells? And the second aspect is what other medications are done to these patients, right? But that is how clinical trials, this is how clinical trials happen. We cannot say what is right and what is wrong immediately. But over the time and multiple people do this and it becomes statistically significant, then probably we can say that these results can be taken further. So I think here the bone marrow is from other sources, okay, and the problem here is in non-union sites. They have taken bone marrow from these same patients. They tried about 60 people, and out of them, 53 have got joined, and that's a remarkable success in a way, right? So you can read about all these studies that are present here, and then we can have an indication about what is right now happening. One I would want you to discuss is, say, for example, why I'm taking a little bit of time here. Yes, there is a patient, and the patient has taken a concoction, right? Like, what do you call that? Some medicines you make. Say a person with COVID has taken concoctions and the person has survived. A person has taken some steroid or the evidence-based medicine and they have survived. A person did nothing or took paracetamol and survived. Which of these is a medicine then? Right? We cannot immediately say which one has worked. And there are also, we are saying only about the survivals. We could also take about how many of this person, if this person has taken concoction but died. There are people who have taken allopathic medicine or evidence-based medicine, modern medicine, they died. Person who didn't take anything also died. Now, which one is a medicine? It becomes difficult, and that is where statistics come. We need to look at a large number of people because there are too many variables in this case. Say out of 15, only 20, out of 20, 15 survived. There are the biology or the genome, genetic predisposition, and many other things about these individuals is also different. We took 20 people, and each of them have different genome, and we don't know how they're going to respond to different treatments, right? So I'm not saying anything about what is a medicine or not, but I want you to pay attention to those kinds of critical thinking about what is working and what is not working. There is also another strategy that we can take. We can use these mesenchymal stem cells as vaccines. That is, you take the mesenchymal stem cells, you populate them, and then we transfect the DNA plasmid into the mesenchymal stem cells. So the mesenchymal stem cell will keep producing the peptides based on whatever is coded by the plasmid that is now in the cell. And we put it into the individual, and the cell is going to produce these fragments that are cloned in this plasmid. So how do vaccines work? We give an antigen, a part, a subunit, or a whole attenuated organism into the body. So the body will produce antibodies against them. And then that is how we are prepared for to clear off any future activities or infections. Here, mesenchymal stem cells are used as those delivery systems. They keep on producing. We don't need to give a booster dose because the mesenchymal stem cells will keep producing the subunits of the pathogen so that the body is primed and the immune system is now active against the antigen as well. So that is one other way of how we can probably use it. The people have been trying these as well. And then there can also be, instead of giving it as a vaccine, you can give it as antigen delivery. That is, you can allow it to produce cytokines. You can allow it to give rise to, this is antigen presentation. That's on top of you have the MHC2, and then they can probably be produced. Of course, these happen to be with modified mesenchymal stem cells. This is what the authors have. This is a compilation of one of the about their bone. How to generate? They are combining stem cell therapy with gene therapy for bone regeneration. So what they would have done is they would put in the vector BMP2. This is the BMP2 protein in a gene in adenoviral vector, and then transfect it into the mesenchymal stem cells. So the mesenchymal stem cells will produce BMP2. They tried it in mouse models, and what they have seen is they have seen that it can produce. It is continuous delivery of osteoinductive factors, that is, those that can induce the production of bone, and that can happen at the site of, and they can repair the bone actually. So the same things can be done in all these cases. It is about bone, BMP. So they have taken a cell, as we have seen here. You can take a cell and they put an adenoviral vector that is secreting BMP. I'm talking in terms of bone regeneration thing. And those cells were given into the body, into the model organism there. And there they were able to see effects of the formation, effects of the bone, repair of the bone, and other things. There will be a lot of clinical trials they do. I mean, these are experiments that people do on live animals. If we are doing experiments with stem cells, it's a very common thing to work with majority of these animals as well. And as you will study in the future courses, where you'll have courses on tissue engineering. You'll also, this is one thing that we are briefly discussed on this today. We will study the tissue engineering, where we want to build the tissues outside the body by building scaffolds. And mesenchymal stem cells happens to be, scaffold is something you make in a structure, three-dimensionally looks like the organ, or even two-dimensional. And then we put the cells, we put the cells on top of them with the culture medium, and the cells will grow on the scaffold and will take up the shape and will become the tissue that we want to. So we should have taken mesenchymal stem cells at least are a good source if you want to make bone-like things or cartilage and other stuff. Then mesenchymal stem cells are very useful. And as I said before, in majority of the stem cell technology, mesenchymal stem cells are the most successful ones of all the trials that we have seen, because ease of obtaining them is first one. Second is they have immunogenic properties. Third is they can give rise to different types of cells very easily. And fourth is they are easy to be manipulated in vitro when we want to do any genetic manipulation and other things. Okay, I'll stop here, and that is the end of chapter. If you have any questions, I can probably try to explain. Any questions? Yes.